Thank you for joining us today for this webinar. My name is Dr. Martin Graf-Utzmann. I'm an editor for Wiley Analytical Science, and I will be moderating this webinar with the title Real-Time Image Feature Extraction Enables a Range of Novel Flow Cytometry Cell Sorting Applications. Today's webinar is brought to you by BD Biosciences, and we're pleased to have Aaron Middlebrook joining us here today as our presenter. Aaron is the BD Biosciences resident application expert for the BD CellView image technology and a long-time flow cytometry expert. He has spearheaded all data generation efforts to evaluate and market the BD CellView image technology. Before starting today's event, let me remind you that this is an interactive web seminar. So you can ask questions at any time during the presentation by typing your questions into the questions box. Only your presenter will see the questions, so don't be shy about asking them. Questions will be answered during the Q&A session at the end of this presentation as time permits. If Aaron isn't able to get to your questions today during the live broadcast, then he will do his best to follow up with you later, offline. So without further ado, let me turn the floor over to Aaron. Aaron, it's over to you.
Thank you for that introduction. So hello, as you just heard, my name is Aaron Middlebrook, and I am excited to have this opportunity to review some of the applications we've done using our recently launched flagship flow cytometer, the FACSDiscover image-enabled cell sorter. I've had the good fortune of being the applications lead on this platform, which means I've had access to this technology through all its different forms of development over the past 5 years. I've learned a lot about what the technology can and can't do, and hopefully over the next hour, I can impart a bit of that knowledge to you. So before I dive into the meat of the applications, I want to talk to you a little bit about what I'm planning to cover in this talk. So we'll review the cell sorter itself and some of its features, and then I'm going to go over how the images are generated and the imaging features, how they work and what they're actually measuring, and some examples of those imaging feature measurements. And then I'm going to give an overview of FACSCourse, which is the software that runs this instrument. We're going to talk a little bit about some of the QC software enhancements and some other features that really impact the user experience. And then I will review a number of applications. I've got an example of translocation and Golgi morphology, mitochondrial morphology, cell cycle, and a cell-to-cell interaction assay. So to start, let's talk a little bit about the instrument, the FACSDiscover S8 cell sorter, and then we'll dive into the image generation portion. So this technology actually was unveiled to the world in a 2013 Nature Photonics paper by our own Eric Diebold, where he described his FIRE technology, as he named it, the fluorescence imaging using radio frequency tagged emission. And from that point, his small company, Omega Biosystems, was acquired by BD, and we've been working for the past 5-6 years developing this technology, and have now packaged it into a sorter. Its sort capabilities were unveiled in a 2022 science paper that I had the pleasure of participating in, and that's a really nice collection of different features and sort capabilities that was actually done on our early access instruments. And so now we've taken that basic same fundamental technology and packaged it into our flagship sorter, the FACSDiscover S8. So all the work that I'm showing today was actually done on this instrument, and this instrument just recently launched over the summer and is now commercially available. And this is a remarkable collection of features. Today we're going to focus on the imaging, but in addition to the imaging, real-time imaging that allows you this mouse over capability of seeing your events populate and images of those events in real time. It also has feature extraction, so some onboard algorithms that will extract metrics of those images and allows you to sort on those. On the other side of it, it's also a very sophisticated spectral sorter. So there's 78 APD detectors and some novel technology in our spectral unmixing algorithm called spectral effects. We can do six-way sorting. There's three nozzle sizes. The setup in QC is all automated and it's all new. No more CS&T beads. So and then there's an onboard LED. So there's a lot of features that are on this instrument that I'm not going to talk about today. I'm going to focus on the imaging aspect of it and what that technology does for you in terms of the applications that it enables you to do. So to talk about the imaging, we need to first understand the imaging. So this is a very simplified, breakdown of how the images are generated, but essentially the images come from a PMT signal. So this is a classic piece of hardware that detects light on flow cytometers. And what we do is looking at that figure on the top left, if you imagine the laser beam, the blue laser, the view you're looking at is the laser is coming right at you. And what we do is we take that laser beam and we split it. And we run half of it goes to the APD array and is for spectral detection. And then the other half is for the imaging. And so we take that blue laser and again, the imaging is only available on the blue laser. So that blue laser is actually run through what's called an AOD or acousto-optic deflector. And that AOD, essentially what it does is through orthogonal acoustic wave pattern, it actually takes the beam, the laser beam, and breaks it up into what we call beamlets. So these beamlets are approximately 1 micron apart. And this array of beamlets is then through some other interference optics, we impart a frequency signature on each one of those beamlets. So if you were looking at the laser beam from the left to the right in this figure, the frequency, the kind of vibrational frequency of that laser, that part of the laser has a signature frequency. So, and then we vary that frequency by an incremental amount as you go across the laser beam. And each one of these frequencies can then be deconvoluted at the other end when we get that waveform pulse off the PMT, and we can use that to figure out what part of the laser the fluorescent signal came from. So as you work your way across that laser beam and you get these beamlets of frequency signature light sources, the cell actually passes through that laser beam, and then you get another variable, which is time. So the front leading edge of the cell breaks that interface of the laser beam, and that is effectively time 0. And then 1 micron later is time 1, and so on and so forth. And as you work your way from the bottom to the top of the cell, you get this line reading effect where you get a different set of time and frequency variables for each part of the cell. And so all this information is encoded in this waveform here, which if you look at the basic shape of that waveform, it looks like a traditional PMT pulse, but those jagged lines are actually all that frequency encoded information that we can extract from that waveform in a process called Fourier transformation. And so out the other end, you end up with an array of pixels, and each pixel has a different address. So that top left one is frequency 1, time 1, frequency 2, time 1, and so that first line is the first line of the cell, second line, third line, and so on and so forth, and allows you to reconstruct a spatial distribution of fluorescence that effectively is a representation of the cell. So it's not a picture of the cell as much as it is an interpretation of spatial frequency or spatial distribution of fluorescence. So once you have the image, and this is a 2D image, and it's derived from that PMT generated waveform, once that image is assembled, then the next step is what we call feature extraction. So that's the bottom half of that slide. And so what you end up with is this series of images, as there's 6 different channels, right? So you have light loss, which is that kind of gray and black image that you see there, which is very much like a bright field image. And then you have side scatter and forward scatter, as well as 3 fluorescence imaging channels. So in this particular example, all I'm showing is the light loss and the FITSI channel, for instance. And in this case, what we do is We actually use the side scatter signal, which I'm not showing here, but that's the one that most adequately reflects the shape of the cell. And so using the side scatter image, we can generate this mask, and the mask is then applied across each of the 6 channels. And once the mask is applied, then these algorithms that are kind of built into the hardware can extract a lot of these image features. So I'll get into exactly what the imaging algorithms are measuring, but for all intents and purposes, these algorithms extract these image features, and then the features themselves can be used to generate plots. And I like to tell people you can't sort on an image, but you can sort on image features that were extracted from images. So that plot there is eccentricity versus radial moment. Those are both imaging features that we'll talk about in a few minutes. And that allows you to pull out doublets. So that green blob in the center is where your singlets are, and you can see from the images there, the top box is your singlets, and then the pink is your doublets, and the bottom right is kind of your, what I like to call your abnormal cells. So cells that aren't necessarily doublets, but aren't necessarily circular and nice singlets. So that's kind of the workflow for how the images are generated. Let's get into exactly what the imaging features measure and how they measure what they measure. So I'm going to talk about 8 imaging features in this talk, and I think there's a total of 12, but a lot of them are building blocks, for instance. So for this first one is called eccentricity. And eccentricity is just a fancy word for kind of circularity. So the opposite of eccentricity is circularity. So eccentricity is measured by looking at a ratio of the long axis. If you look in that middle column there is kind of the mathematical explanation of what's being measured. So that red or long axis divided by the blue or short axis. So you also get these imaging features, the long axis and the short axis, that you can actually look at on the instrument as well. But they were building blocks to get to eccentricity. So and eccentricity can be used for looking at the shape of a particular fluorescence pattern. So a very eccentric shape would be something that's very long and linear, whereas something that has very low eccentricity would be circular. And you can see that from those images on the right. And doublets, for instance, have a very high eccentricity. They're very long and narrow. So this is a good feature to pull out doublets. It's also used in a lot of different cellular morphology assays, and then of course cluster and doublet identification. The next one is radial moment. So radial moment is essentially what it does is we're in all these images we're looking at the green signal. So in this case, I think this is a GFP. I think it's GFP of mitochondria, if I'm not mistaken. But essentially, the radial moment measures the median distance of all the pixels from the centroid. Now the centroid is the relative center. So in this case, what is the center of the green signal? So in the little schematic there, the explanation, we have 2 green dots, and the relative center of those dots is kind of halfway between the 2 dots. And so then what it would do is measure each pixel that has a green signal in it and how far that is from the centroid. So essentially, it kind of gives you this idea of spreading. So something with a very low radial moment would be those cells on the left there that have kind of a concentrated fluorescent signal that doesn't extend to the outer portions of the cell, whereas something with a really high radial moment would have a lot of the fluorescence kind of populated around the surface of the cell or the edge of the cell. So a donut has essentially the highest radial moment, or like a surface stain would have a really high radial moment, and then a nuclear stain would have like a really low radial moment. So this is also another one that's used in cluster identification, because things that have a high radial moment, doublets have a high radial moment because of where that center falls between the 2 cells in a doublet. So cluster identification, doublet discrimination, and then also a number of different cellular morphology assays utilize radial moment. So the next 2 are size and total intensity, and I always put these together because they're similar in a way. So size is actually just a count of all the pixels that have that green signal present. So you can control how liberal or conservative you want to be when you set that threshold, that mass threshold that we talked about. So that's a user controlled function on the instrument. And if you want to have very stringent size metrics, then you're going to be very conservative with that threshold. And if you want to be more liberal with it, then you have that option too. But essentially, once you set that threshold, that determines how bright of a signal will be counted as a positive signal. So in this case, size is just the number of pixels where there's a green signal present. And much like that, total intensity uses that same pixel count, but now it asks the question, how much fluorescence is in all those pixels? So the size part is kind of binary, how many pixels, and then total intensity is if you have some pixels that are really bright and some that are really dim, then they'll be the additive intensity of all those pixels. And they track very closely. So in this case, there's not a huge difference between size and total intensity, but that's not always the case. So depending on what your application is, so size is a really good one for label-free sorting because you can look at side scatter and forward scatter size. And then, of course, cellular morphology is almost in all of these can be used for morphologies of different natures, depending on what biology you're actually looking at. The next two are correlation and delta center of mass, and these I put together because they are somewhat similar. So these both use you have to have more than one fluorescence channel. So in this case, we're looking at, in both cases, the red and the green. So this is the correlation is going to look at what's the overlap between two fluorescent signals. So in this case, the red and the green channel, I always think of them as FTSE in the green, and the red is like a nuclear stain like DROC5. So in this case, those pixels where the two signals overlap, that will be assigned a score based on the percentage of pixels from those two channels that occupy the same space. So as those two signals become more and more co-localized or occupy the same space, that correlation value will go up. So in the case on the right there, we have, this is a nuclear translocation assay, and those images on the left where you see green fluorescence and red fluorescence kind of discretely occupying different parts of the cell, that's a green cytoplasmic stain and a red nuclear stain. And they're pretty highly uncorrelated, so they don't occupy much of the same space. Whereas once that cytoplasmic stain moves into the nucleus, we see more of a yellow color, and that's the green light and the red light mixed together to make yellow light. So those images on the right are highly correlated. And now delta center of mass is similar, but slightly different. So what this one does, it measures the distance between the centroid of the two channels. So in this case, the centroid or relative center of the red fluorescent signal is measured against how far away it is from the relative center of the green fluorescence channel. So in this case, you have the two green dots and the one red dot, and the relative center is the distance. That blue line represents the distance between those. So this is really good for like intracellular trafficking. You can actually measure whether something's, like if you have a nuclear stain and you have a viral particle, or maybe a pathogen, or even a metabolite that has a fluorescent signal on it, you can track it as it goes from a passive binding to the outside of a cell to the inside of the cell, and then closer and closer to the nucleus. So you can track the distance between two fluorescent signals. So in this case, you can see that green and kind of fuchsia color particles. The ones on the far right are actually coincident cells that went through the cytometer through the interrogation at the same time. And then those ones above it are actually closer even, and then the ones on the left, it looks like in some cases the green particle has been internalized by the cell. So as you get two signals close together, their delta center of mass goes down. The next one I'm going to talk about is max intensity, and max intensity is fairly straightforward. It's just the single brightest pixel in any given channel. So this is great for punctate fluorescence. I use it in phagocytosis assays and cell cycle. it just essentially measures the brightest pixel. And so you can see there that, the cells on the left that have a low max intensity are very kind of dim, and the ones with the high max intensity are extremely bright. And then diffusivity is somewhat related in that it uses max intensity, but what this is the measurement of the total fluorescence, the area divided by the max intensity. So this kind of looks at, okay, how much of that total signal is being driven by that max intensity. So if you have a very few number of pixels that are all kind of the same brightness, that fluorescence number on top is not going to be very big, but that max intensity is going to be very big. So you'll have, low diffusivity. Whereas if the opposite is true, if your fluorescence number is quite large, but your max intensity is low, then you're going to have a high diffusivity, which means the signal is very spread out or diffuse, essentially. So this is also another one I use for phagocytosis. It's also a really good one to look at when you're doing GFP sorts. If you have a GFP signal that's maybe not as bright or centralized as you might want it to be, we get a lot of false positive GFP sorts because of high background or diffuse staining. And this will allow you to maybe clean those up a little bit. And I'll show you an example of that's somewhat related to that. Um, So that's pretty much the imaging features and how they're measured. And again, I think there's a total of 12. Those are the main imaging features. All the other ones that I didn't talk about are building blocks to get to those 8. So the next thing I want to talk about is the FACS Core software and some of the features that we've implemented in that software. So if Fax Course sounds familiar, it is the same software that we used on the Melody, but this version that we've implemented for the Fax Discover is quite a bit different than the Melody version. The base of the software is the same. It has the same basic layout with kind of those arrows across the top that give you a guided kind of workflow. And there's a number of other pages, but I'm going to focus on the view data page, which is kind of where you do most of your work and where you see the acquisition of your data. So the layout is underneath the arrows for the guided workflow. You have, all your instrument controls in terms of, how many events you want to save, whether or not you want to save images, which images you want to save, and then of course the control of the instrument, loading tubes, unloading tubes, recording, and then flow rate, of course. And then on the left there, you see your data sources. So those are your different samples. So I've recorded a number of samples in this experiment. Your gating hierarchy to the left there. And then in the center, the top part is all your plots. And those plots can be, resized and rearranged any way you want them. And then there's a spectral plot down below that gives you, the raw data for each of your 78 detectors as well as your imaging detectors. And then over on the right is kind of the showpiece of this new version of Chorus, which is the image wall. And one of the nice little features of the image wall, in this case, I'm looking at these PDCs and I have my mouse, hovering over one particular cell. And as I hover over that cell, that event actually will show up in all my plots. So where is that event? It really helps you kind of find your cells. It helps you make that connection between what the image of your cell is and where it shows up on your plots. Um, and then if I scroll through this, there's a couple controls in here. If you see that image wall on the right has changed as I went to the next slide, but that allows you to, control what imaging channels you're looking at. So in this case, I'm only looking at one fluorescence channel and the light loss channel. So I have forward scatter and side scatter turned off. This is also where you control for that threshold that I talked about. And gives you some image controls, which are very similar to other microscopy or even Photoshop image controls, where you get this histogram and then a max and a min, as well as a gamma control and some smoothing features, and all that is essentially cosmetic because it doesn't affect the actual data coming off the instrument. It's just how the images are shown, how bright or dim you want those images to be. So you have that granular control of each channel and then you can control which channels you want to see or not see. Um, so as a just as a sorter, if you don't think about the imaging in terms of how it impacts what applications you can do. How does the imaging just help as far as as a sort utility, for instance? So the one thing that I always come back to is the mouse over aspect of it. So to be able to, see exactly what cells are that you're gating, it really becomes intuitive as you use this instrument more and more, and it guides almost every gating decision you make on some level. And then, of course, some people don't like it, and we have the ability to turn it off if you find it distracting as you're drawing your gates. But I've really gotten used to it, and it's a big help. And then, of course, the imaging features themselves, just with the light loss alone, or with side scatter, I can really pull out doublets and really clean up my sorts and really have a lot more confidence in the gating that I'm doing. And then I always like to showcase this bottom part because for people who do index sorting, it's really a revelation. You can index sort up to 100 cells per well, and with this user interface, you can actually click on, once the sort is complete, you can click on a well and you can see all the images of the cells that went into that well. And not only that, it'll show you, like in this case, I've selected, well A11, and there's 10 cells in there, and you can see in the plots to the right, it has this washed out effect so that you can appreciate where those events actually came from. So there's green dots that highlight the 10 cells that are in that well and what part of each plot they came from. So it really gives you a sense of, allows you to clean up your gates if you need to, if you're getting some missorts, if you're getting cells that don't have the level of fluorescence that you want, you can tighten up your gates and see where those outliers are coming from. And then, of course, it really gives you unparalleled QC ability, so you can go through and look through an entire plate and see if there were any missorts and make a note of say, which wells didn't have a cell in them, or maybe they had two cells or the wrong cell, and you can, make a note to maybe not sequence those, or don't proceed with whatever downstream application you're going to do for those wells. So really nice way to QC your index sorts. Now, I'm going to actually go over a few other chorus features that I really like. So I already talked about the mouse over part, which is a really important feature in this technology. But then there's other ones that we've included. So there's this scroll wheel driven plot zooming. And so what you can do is you can hold the mouse over the x-axis or the y-axis and use your scroll wheel, and you can expand or contract that scale. And then you can also zoom in to a plot and actually look at each of the dots at a very granular level. So that it really allows you to mouse over really dense populations, and then come zoom back out and get the plot to where you had it before. So that's also another nice feature. And related to that is actually this other feature, which is very similar, but it allows you to really scale so this is a bi-x plot, and these arrows at the top allow you to get your bi-x scaling exactly where you want it, and then you can actually pick up the whole plot, and you can stretch it and mold it to fit that space that you want. So all those bi-exponential editor dialog boxes that you may be familiar with from Diva or a thing of the past when it comes to this platform now. So I find it takes a little getting used to, but once you start using it, it's a lot quicker and intuitive, I think, than previous editions. So let's transition to the applications part of the talk. This is kind of a laundry list of applications that we feel might benefit from imaging, either make it a little easier to do these type of applications or enable a new category of applications. I'm not going to talk about all these applications today. I've chosen a subset. The first one we're going to talk about is nuclear translocation. So nuclear translocation is a kind of a classic imaging, application that it's a really nice example of an assay that you wouldn't necessarily be able to do Well, you wouldn't be able to do it at all on an instrument that can't tell you information about spatial distribution. So in this case, what we're looking at is NF-kappa B and DNA as your two signal sources. So the green there is, FTSE stained NF-kappa B P65 pan-in kappa B antibody that I'm looking at. And then in the images on the left, you see the NF-kappa B distributed in the cytoplasm. And then I have a nuclear stain. In this case, I have DROC5 in the red there that stains the nucleus. And so, and if you look at the top there, you see a nice, distribution of fluorescence staining in the FTSE in the green cytoplasm, and then the DROC5 in the nucleus there. And as you treat these cells with TNF, that NF-kappa B effectively translocates into the nucleus. So in that intermediate stage there, you're starting to see some of the NF-kappa B has made its way into the nucleus, and you see that it shows up as a bit of yellow in the nucleus. And then once all the NF-kappa B has fully translocated into the nucleus, you see it going to completely yellow. And that's reflected in those histograms at the bottom, which is essentially the correlation score. So that correlation, the median of that correlation peak on the left is at or around 0. And then as you move towards the translocated version, that median gets closer to 1. So something that's fully correlated would have a correlation score of 1. So if we look at the data for this experiment, what we're looking at is just a couple cleanup gates. We've got a scatter gate there on the top left, and then some doublet discrimination gates. The one in the middle there is your classic height versus width gate to exclude doublets, and then we use the imaging parameters of eccentricity and radial moment to further narrow down our gate to singlets. And what you see there is a mixed population of translocated and untranslocated cells. So you can see in that image wall shot at the top there, you see kind of a yellowish green nuclear stains where the NF-kappa B is fully translocated, and then a number of cells where you can see a discrete nucleus in red and the cytoplasm in green. And so that's a heterogeneous population. We actually sorted those out onto a microscope slide and looked at them, and what we see is exactly that. So you see some cells over there on the right that are translocated and some that are untranslocated. So what we did was we went ahead and sorted those two populations. You can see the sort gates there. I've sorted the uncorrelated ones and then, of course, the correlated ones. And then we look at the sort products, and we actually run those sort products through the instrument again just to check for purity. And what we see is on the top, they're largely uncorrelated, and you can see from the image wall, you see nice discrete green and red signals in those cells. And when you sort them onto a microscope slide and look under the scope, it's confirmed they're all untranslocated. And then the converse is true with the correlated ones. So all the images on the image wall on the cytometer, you get nice greenish yellow nuclear stains where the NF-kappa B is translocated into the nucleus, and then that's confirmed on the microscope. But the reason I'd like to show this slide is it really brings up one of the things about imaging that's unique that I think users or flow cytometry users are going to have to get used to, which is this thing called orientation effect. So in this case, if you think about the difference between an uncorrelated version of the cell and a correlated version of the cell, on the bottom there, no matter how we look at that cell, so in this case, the cytoplasm is translucent, and you can see no matter what direction or what orientation that cell went through the cytometer on the second time through when we did our purity checks, it still stays essentially totally correlated. And that's because no matter what orientation you look at that cell, those two signals occupy the same space. Now, it's a little bit different for the uncorrelated version. So the uncorrelated version is a little bit like looking at two balls of signal. And if they go through the cytometer in an orientation that allows the imaging feature of correlation to be measured really, uncorrelated. You get nice green and nice red separated. It's going to be classified as an uncorrelated event and will be sorted. Now, when we reacquire that event, because of the nature of those two things being next to each other in one orientation, if you turn that cell 90 degrees and you actually look at it from, kind of an axial perspective, those two events, those two signals might seem to overlap a little bit. And so you'll see that CV kind of inching towards correlated. And what that is just a small proportion of my cells that went through at an orientation that made them look a little bit correlated. And it's not that the instrument is not sorting properly or is things are drifting out of your gates. I think in flow cytometry we're used to sort products drifting out of gates due to some MFI drop that you see on repeated acquisition. But this is something a little bit different, and you really need to understand your biology going into this. Usually, imaging features that measure two fluorescence channels, like delta center of mass and correlation, tend to be, this kind of artifact can arise, but it's not the biology and it's not an artifact. It's strictly orientation. And I'll show you a number of imaging features where this doesn't happen. And that's because the orientation doesn't matter. A lot like the correlated ones, orientation doesn't matter on that because the location is the same no matter what angle you look at the cell. Next, I'm going to talk about this Golgi morphology application. So this is kind of an interesting effect that happens. So what we have is we have some HeLa cells that express GFP in the Golgi apparatus. And when you treat cells with Berfeldin A, what happens is, for a lot of you who've done intracellular cytokine staining, you'll treat your cells with Berfeldin A because that blocks the secretory pathway and you get this cytokine buildup in your cells. And the way it does that is it actually disrupts the Golgi, and the Golgi then gets redistributed throughout the endoplasmic reticulum. So the effect of Berfeldin A on a GFP expressing HeLa cell in the Golgi is that untreated cells have this very kind of punctate expression, and you can see the images there at the bottom. Untreated cells have this, discrete Golgi expression, and then once you treat with Berfeldin A, you see this dispersion of the GFP Golgi proteins throughout the ER and as well as the Golgi. So we can kind of use this effect to look at different spatial distribution of fluorescence. And on the cytometer, that allows us to actually sort these cells. So if we look at those gates at the top, another nice singlet gate, and then the next plot is looking at the GFP positive cells, and they show up as one continuous population. But then what we can do is we can look at a couple different imaging features, in this case, diffusivity versus max intensity, and we can actually pull those two populations apart. So the diffusivity, again, measures the total fluorescence divided by the maximum intensity. So those cells that have a broad distribution of signal have a higher diffusivity, and then those cells that have that kind of, punctate expression of the Golgi have a lower overall fluorescence and a high maximum intensity. So you can't really differentiate these cell populations just on the fluorescence alone. They overlap almost completely depending on how bright or how broadly distributed that dispersed signal is. But using the imaging features, we can easily pull these two populations apart. And then, of course, the sort purity checks there down below. And then we also sorted those onto microscope slides, and you can see that morphology a little bit more clearly on the microscope, but it's very similar to the images you see on the cytometer. The next application I wanted to talk about is this mitochondrial morphology assay that I did. I wanted to see if I was a classic, T-cell immunologist, how would the imaging impact my work, and how might I leverage imaging in the context of smaller cells? Like I've shown you a lot of bigger cells so far, HeLa cells, and the first cell type were HT 1080s, which are both very large cells comparatively speaking. So what I wanted to look at is mitochondrial morphology in the context of T cell activation. So does it change as these cells get activated, and is there any correlations I can make from activation state or surface cell markers to this any morphological or changes in mitochondria that I might see. And so what we did was we took some PBMCs, naive lymphocytes, and we stimulated those for 48 hours with CD3, CD28 dynabeads. And then after the course of 48 hours, we looked at, unstimulated versus stimulated, and we did a few different surface antibodies as well as mitotracker, which will actually stain the mitochondria, and we can look at mitochondrial morphology of these cells. So here's our panel we had. So we have CD4, CD3, CD45RA, CD197, CD127, CD25, and CD69, of course, our mitotracker. And just overtly looking at the flow plots, the green box in the top there is your unstimulated cells. And then the red at the bottom is your stimulated cells. And so, the biggest differences we see are obviously that last plot on the right, looking at CD69 and CD25. And as these cells become stimulated, you get a massive upregulation of CD69 and 25, this large population of 25, 69 double positives. And now when we look at that in the context of mitochondria, we can look at those same two populations. And looking over on the far right, we're looking at the radial moment of the mitochondria. So that's how much spread from the center. So I imagine if I also looked at size, I'd probably see an increase as well. But the radial moment seemed to capture this bimodal distribution of intensity or of spread of the mitochondria. So once I saw that, I wanted to kind of dive a little bit deeper into what exactly I was looking at. So what I did was on the unstimulated, I looked at cells in both those gates, the low radial moment and the high radial moment. And while there were very few cells in the high radial moment, there are some there. And they do show, that there is some cells that have this kind of signature spreading of the mitochondria, even in unstimulated resting cells. But then when I looked at the stimulated version, what I see is kind of interesting because clearly that high radial moment population is corresponding to the CD25, CD69 double positive cells. But then when I look at that low radial moment side of that histogram, I actually see there's still quite a large proportion of 25 and 69 positive. So it's not absolute, is what it's telling me, that there are a population of activated T cells that don't undergo this hallmark mitochondrial morphology. So it was an indication that there's maybe a little bit more biology there. I dove a little deeper and looked at kind of the heart of that 25-69 double positive population and looked at diffusivity versus max intensity and was able to pull out kind of a range of different morphologies. There's really these bright spread out versions and then more of a dim diffuse version. And whether or not I'm looking at just the spectrum of activated radial moment of mitochondria, or I'm looking at discrete different phenotypes, I don't know. And then the other thing I noticed was even when I look at all those 69 negative or 69 positive, 25 negative, and 25 positive, 69 negative cells, I see a range of different mitochondrial morphologies. The underlying kind of message here is that the imaging, even in a small cell case like activated T cells or activated lymphocytes, it gives you an additional layer of interrogation. I think this is just the tip of the iceberg in terms of what this mitochondrial morphology is the biology behind it and what role it plays in activation. It's clearly a little bit more complicated than just activated cells have more mitochondrial activity. That seems to be the driving trend of the biology, but there's clearly subsets of cells that have a activated phenotype from a surface marker perspective, but a radial moment or mitochondrial morphology consistent with a resting or unstimulated T cell. So there's further populations to explore and just another layer of information that you have access to when you engage this technology. I also wanted to talk a little bit about the cell cycle application. So this was an application that we showcased in the science paper, so I'm not going to go that much into detail, but I think it's a really nice example of using several different imaging features and fluorescence features to identify, these very, visually striking populations. So what we did was we had some HeLa cells expressing a neon green in one of the histones, H2B, and then we also had an Elixir 647 stain on histone H3, and then DAPI, as well as a surface stain there in the red. We use CD147P CF594. The gating strategy is a little complicated, and I'm not going to go into the full depth, but essentially what we did was we used 3 different fluorescence parameters in conjunction with 5 different imaging features. So the imaging features there are radial moment, H2B, the neon green, max intensity of the neon green, eccentricity of the neon green, and then of course eccentricity of forward scatter and side scatter. So what you're doing is you're using that shape of, that characteristic shape of almost a doublet for the anaphase and telophase portion of it. So that's where we use the eccentricity of forward scatter and side scatter to really pinpoint those cells that resemble doublets. And then, of course, the metaphase plate has that kind of linear distribution of fluorescence across where the chromosomes are lining up, and that allows you to pull those cells out. So let's look at the the images off the cytometer and compare those. So using that gating strategy outlined there, we can really pinpoint each one of these 5 populations, and what you're looking at is um, the images off the cytometer, the image wall, and then we actually sorted those onto a microscope slide and looked at the images on a slide, and they match up quite nicely. So a really nice demonstration of using both fluorescence and imaging to really target different populations. The last one I want to talk about is this cell-to-cell interaction. So this has become a little bit of I wouldn't say popular, but it's an emerging area of research. A recent paper by Julie Burrell outlined these T-cell monocyte complexes and how they're markers of immune perturbations. And so we decided to try and take a look at this using the imaging features and how they help us identify these cell populations. So what you're looking at here is that's actually a sorted version of this T-cell monocyte on a microscope. And so we've got the green in CD3 and the cyan in CD14, and then a dot plot there of 14 and 3, and you can see those double positives. I think for a long time people thought they were artifacts. I think there is some that are an artifact that we found are more coincident doublets, and then there's some that are actual complexes. So we wanted to ask the question of, does the imaging help you identify these cells, and can you use those imaging features to sort this population? So that's exactly what I did. So in this case, I have some additional markers. I've got CD45 in here and also CD19 because I also wanted to look at B cell T cell interactions. But essentially, we have that scatter gate on the top left, and then the doublet gate in the middle there. And then we gated on 45 positive cells and then CD19 negative cells, and then that CD14 versus CD3, and looked at those double positives. And what we found is there's kind of several populations of doublets in there, but the 2 basic ones are coincident doublets. So that's where the green or T cell and the cyan or monocyte went through the interrogation at the same time. And you can see they're separated by some space there. And then, of course, you have the ones that are actually touching. So those are cells that may be engaged in maybe some communication. So the ones you want, or the ones in this case that we wanted, were those ones that were engaged. So using radial moment and correlation, so that's again the spread, so how far apart things are spread out, and then correlation is if there's a bit of overlap between the cyan and the green, I'll get a good correlation. So I can look at those coincident versus what I call synaptic doublets, and we can actually sort those. And so we sorted both populations. First, we sorted the the coincident doublets, and as you might expect, those coincident doublets, when you reacquire them, they essentially are all singlets. And we actually sort those onto a slide, and of course you get a bunch of isolated monocytes and T cells. Now, conversely, when we sort the synaptic doublets, what we see is a lot of them actually survive the sort. So you can see there's some that redistribute in CD14 positive single positive cells and CD3 single positive cells, but the majority in this case are CD3, CD14 positive, and they actually remain intact. And we actually can sort those onto a slide and see our little kissing cells there. So that was kind of remarkable. I did see that this was a little bit donor dependent. Some donors, their T cell monocyte complexes really stayed together and survived the sort. I also saw that when I did this with B cells, the B cell T cell complexes almost inevitably didn't survive the sort. So there's something special with these monocyte T cells that gives them a little bit more of a stronger affinity and survives the sort. And you can actually see that here with the B cells. This is a B cell and a T cell, kind of very similar gating strategy, but when I sort these, they don't survive the sort. So I think that's all the time we have today. So I'd like to thank a number of my colleagues that made all this work possible and really helped bring this instrument to market. And if that's all, I will take some questions.
Aaron, many thanks for your presentation. Before we start with the Q&A session, I would like to ask you to fill out the poll that has been sent out via the chat. Um, we have a little time for the question and answer session. If you haven't submitted your question yet and would like, now would be the perfect time to do so. Let's go ahead and see what questions we already have so far. So, what are the cells stained for in the FIT-C?
I think when that question came through, if I remember the slide correctly, it's actually the same assay that I outlined in the Golgi treated with Brefeldin A. So you had a very punctate expression and also that kind of dispersed expression. So it's a HeLa cell expressing GFP in the Golgi apparatus.
Okay, thank you. And an add-on question to that would be, can you use traditional flow cytometry dyes for this kind of imaging?
Yeah, so for all intents and purposes, all your traditional flow cytometry fluorochromes work well with imaging. The only requirement is that they have to be excitable off the blue laser, so you are restricted in that sense. And there's 3 imaging channels, and so depending on which fluor you pick, you have to pick fluors that obviously don't fluoresce in the same channel at the same time. But your traditional fluors like PE, FITSI, P size 7, all those work just fine with imaging.
Okay, so as you said, the imaging part needs to work with the blue laser. So the imaging works with the blue laser. How many colors can be visualized on the blue laser on the S8?
Uh, so right now there's 3 imaging channels, so you can visualize up to 3 different fluorescence markers at the same time. Um, and what I try to do is when I'm using, like if you have 2 surface stains, they're obviously going to overlap quite a bit. So I try and spatially separate those imaging channels. So I might have one cytoplasmic, one surface, and one nuclear, or one Golgi and one nuclear and one surface. You can do multiple at the same location, but when you look at both of those at the same time, it's really difficult to tell when you get two fluorescent signals on an image that overlap in the same space. It either comes across as a mixture of those two colors, or sometimes it looks like it's just white light. So depending on what you're looking at, I try and spatially separate those imaging channels.
Yeah, we've seen that with the white light, with the green and red fluorescing, with the green and red fluorescence that gave off the white light. That was
Yeah.
shown in your presentation. Um, Now, what is the image resolution?
So the actual resolution is approximately 1 micron. So if you have something that is 1 micron in size, you can differentiate between two particles that are 1 micron each. If they're next to each other, you can see that it's two. But once you get below that threshold of 1 micron, it'll actually just show up as one. So from a microscopy standpoint, it turns out to be about 16x is the magnification, and 1 micron is kind of your resolution there.
Okay, thank you. Um, are the photos in quotation marks taken randomly inside a gate that you pre-select, or do you choose the number of photos that you want? How does this imaging bit work, the actual creation of the images?
So in the images tend to populate in the order that they were acquired. So if you have a gate that you want to see all the images for each of the events in that gate, only those images will show up on your wall, and they'll be displayed in the order that they were acquired. And so you can kind of scroll through your wall and look at all the images. And the same thing is true if you're looking at your data in real time. You'll see the cells kind of streaming along on your image wall, and they'll populate in the order that they were acquired. You do have the ability, like I said, to drill down into a particular gate and only show the cells from that gate. And then that's just what's displayed. When you talk about the images that are actually stored, you can control that. You can control how many images you want to save, which images from or which gate the images you want to save. You can't select particular cells within a gate to save. You have to select an entire gate. So, like a target gate. Like if you want to look at Tregs, for instance, you would save all the cells from that Treg gate, and they would populate the wall, and they would be saved onto the hard drive. So that's the control you have over that.
Okay, thank you. Um, can the images be exported in an automated way? The way you described it, they're probably automatically named with I mean, do yeah.
Yeah. So essentially, what happens is the images are stored in a proprietary file format called the CVW file, which is a cell view waveform file, and that allows you to get your images off the instrument in a much quicker fashion. And then there's another step after you get your data off the instrument. We have a utility called an image extractor. And what this is it's a small piece of software that I keep it on my laptop. I have it on all my workstations. And essentially what this does is this will extract the TIFFs from that CVW file. And when it extracts them, it actually puts them in folders. And each folder has 10,000 images, and each image has an event ID that is used. So if you can take this data and you can look at it. We have a plugin for FlowJo, and that plugin will reference those images by event ID, which is part of the title of each image, and you can look at your data offline that way. So yeah, the utility packages those TIFFs into discrete folders, and each folder, like I said, is I believe 10,000 events.
Okay, thank you. That sounds great. Um, so I mean, you showed that the software already has a lot of features to offer, but are you planning on access additional imaging features with future software updates?
So because the imaging features are actually part of the hardware in terms of it has to be done at the firmware level. So there would be a potential possibility to add more imaging features at a later date. It would require a service visit because the software and the firmware would both have to be updated. We don't have any new imaging features planned in the short term, but that possibility does exist. It wouldn't require an actual like new piece of hardware or anything. So that potential is there. And at BD, we're really committed to increasing the utility of this instrument with future software updates. So that possibility is there.
Okay, so I think we are kind of also running out of time. If we don't get any new questions from the audience, um, yeah, I would like to thank everyone for attending this webinar today. I would also like to thank BD Biosciences for all their support with this event, and of course, a special thanks to you, Aaron, for your presentation to all of us today. Um, as a reminder, this webinar has been recorded and will be available for viewing in a few days. Uh, and so this concludes today's webinar. So from Aaron and me, Martin, it's goodbye.